and thank you for the invitation. It's great to be back here at Stanford. So I've actually added in a little bit to the title there. I've added in some proteomic data as well, so I'll show a bit of extra data compared to what I was asked to present. So I'll start off with the metabolomics. Metabolomics in general, the approaches are able to identify thousands of low molecular weight metabolites in biological samples, including urine, plasma samples, and tissue samples. The data we've looked at so far is predominantly plasma data, so that's what I'll present. Metabolomics has already shown translational potential, so there's the intelligent electrosurgical knife that's being developed at Imperial College, which is an electrosurgical knife where it actually collects the metabolites that are being burnt off the tissue as it's being cut, and they can use the signals produced by that to identify whether the tissue is cancerous or non-cancerous to make sure whether they've taken out the whole of the cancerous tissue. In terms of pulmonary hypertension, there's already been substantial publication on altered metabolic pathways in pulmonary hypertension. Specifically in circulating levels of metabolites, we've seen altered tryptophan, purine, TCA cyclointermediate metabolites related to severity of PH, specifically hemodynamic measures. We've also seen from Evan and colleagues the increased acylcarnitine levels, which we were able to reproduce in our study. And then there have been some studies in lung tissue samples as well, which have shown abnormal oxidation, arginine and sphingosine pathways as well. And also from the FDG PET uptake image, we can see that we get increased uptake of glucose into the right ventricle in pulmonary hypertension. So a metabolic shift has been identified there as well. So our hypothesis was very general. It was just that plasma metabolites can distinguish PAH patients from controls, but we'll also be able to predict outcomes, including response to therapy and survival in our patients. The methods I'll be presenting today are based on a company called Metabolon in Durham, and it's a UPLC mass spectrometry, which can identify thousands of different metabolites. So it's an unbiased method as well. So they profile the samples, and they provide data on all of the metabolites that they were able to detect. So it's not from a sort of defined library, although they have a library of metabolites they are able to identify. So we did discovery on a group of patients from Hammersmith in London, and we also had 2 validation cohorts, which was another cohort from London of historic samples that we collected over the last 10 to 15 years. And we also had a validation cohort from all the centres in the UK, the PAH cohort collection, in which we validated our findings. We had 2 types of controls. We had healthy volunteers, which included myself and other people working at the hospital. But we also had disease controls, which are an interesting population, which are the patients that get referred to the PAH clinic, but are found not to have pulmonary hypertension. So potentially a much more applicable group of patients to compare with. And then in total, we had 365 patients with idiopathic or heritable pulmonary arterial hypertension. So the first thing we wanted to do was to be able to show this signature of pH in our patients, and for the metabolites that we're interested in, we wanted to make sure we're only looking at very high quality, well-quantified metabolites. So we only included those that were detected in at least 95% of subjects. And we're only interested in biologic metabolites, so we excluded xenobiotic signatures. We were able to show from an initial analysis of around 1,400 metabolites that 53 were significantly changed in PH patients compared to healthy controls in all 3 cohorts after bonferroni correction. We were also able to show that this was independent of confounders such as BMI, creatinine, bilirubin, so liver and renal function. And then we went on to show that 20 of these metabolites were also distinguishing these patients from the disease controls. So the patients that were referred to the same service could actually be distinguished from those that went on to get a diagnosis of PAH, again, in all 3 cohorts. So just to show you a bit of that data, this graph shows our top 20 metabolites, which are all normalized to the average level of healthy volunteers. So the bars going to the right are metabolites that are higher in patients, and the bars going to the left are ones that are lower in patients compared to healthy controls. The orange bars are the disease control, so again, those patients that don't have pulmonary hypertension, and then the red bars are showing the levels in the PAH patients. This is our discovery analysis for those 20 metabolites, and then the validation analysis, you can see the metabolites are going in the same direction with a very similar fold change of these metabolites. And again, you can see that these metabolites are quite different from the disease controls in orange. Although you can see for some of them, the orange bars are going in the same direction, so there maybe is a trend for the same alterations in disease controls, just not to the same extent in the more severe diseased PAH patients. This is just a quick slide to show that we did have a look within our PAH population as to whether the BMPR2 mutation carriers, pathogenic mutation carriers, actually had a different profile and in the patients that we looked at. And this, the genotype was taken from the study that Nick just presented. We actually saw no real difference in these metabolites between our patients with and without BMPR2 mutations. So we wanted to see if we could create a model using a minimal set of metabolites to actually discriminate PAH from our controls. So we actually did a logistic regression analysis to reduce it to 7 representative metabolites. I say representative because we had multiple metabolites from similar pathways, so we had lots of modified nucleosides. In this case, we picked N1-methylenosine, but we had others as well. We selected a single acylcarnitine, a steroid, and sphingomyelin, but from all of those groups, we had multiple metabolites going in the same direction. So this is the discriminant score that we were able to build using those 7 metabolites. And you can see in blue our healthy controls, in red our PAH patients. So the first 2 are our discovery analysis, and you can see that the score of just 7 metabolites was able to distinguish the patients very accurately. Now in green we have an interesting subgroup. These are the vasoresponders, so the patients that respond to calcium channel blocker therapy and go on to have very good outcomes. Their levels of these metabolites are actually very similar to the healthy controls. These patients are already on the calcium channel blocker therapy, so it'd be really interesting to see as to whether they already had normal profiles at baseline or whether the treatment actually triggered this change. So anyone who has a collection of those would be really interested in looking at that. And then you can see on the right that the 2 validation cohorts really reproduced that very accurately. So you can see in the ROC curve here that we were basically able to distinguish a control and a patient on average 95 to 94% of the time in the 2 validation cohorts. And obviously in the discovery it performed very well, as it always does in discovery. We also had a look at survival, and in the first 2 cohorts, we had a significant number of deaths because of the follow-up period. At the point where we actually did these studies, the 3rd cohort hadn't had many events, so we didn't do a survival analysis in that, but that is something that we will do as the cohort ages. So we had 28 and 25 deaths, respectively, in the 2 first cohorts, and we found that 63 of the metabolites were prognostic in both cohorts, and almost half of those were actually independent of established prognostic factors, including NT-proBNP, 6-minute walk distance, and red cell distribution width, something we've previously shown is closely linked to survival in our patients. And this Kaplan-Meier curve on the right just shows that if you pick a level in the discovery cohort, it's able to distinguish survivors and non-survivors quite accurately in the validation cohort. And again, this is another one of those modified nucleosides. These modified nucleosides are very specific to tRNAs, so you only see these modifications in transfer RNAs. And there are a couple of different reasons why you might see modified nucleosides in circulation, because obviously tRNAs you wouldn't expect to be released normally. And 2 of the scenarios are either a stress response, in which you see increased production of angiogenin, which I've shown here by the little scissors, because it cuts up the tRNA molecules. And once the tRNA molecules are cut up, they are then released into the circulation and degraded into the single nucleosides, which we detect. The other example is in proliferation. So for example, in cancer patients, you also see it because there is a general upregulation of the machinery, including tRNAs for reproduction of the cell. And then you just have an increased number of tRNAs in general, and this normal level of cleavage, so normal levels of angiogenin, will lead to increased nucleosides in the circulation. So to investigate which pathway might be more relevant in PAH patients or whether stress response is relevant, we looked at angiogenin levels in the same patients that we measured the modified nucleosides. So we did this by an ELISA. So we can see here in red again are the patients, in orange are the patients over 70, which we looked at separately, and in blue are controls. But what you can see overall in the population is there is a good correlation between plasma angiogenin levels and plasma modified nucleoside levels. And this is suggesting that part of it is a stress response where you're getting upregulation in angiogenin, which is leading to increased cleavage of tRNAs under this stress. Bioenergetics is obviously of great interest to anyone interested in pulmonary arterial hypertension. So what we normally see is various different sources of energy feeding into the TCA cycle. And what we're seeing in our IPH patients is a buildup of all of the precursor metabolites, including pyruvate, acyl-carnitines, and glutamate. So they're not feeding into the TCA cycle, and this is compatible with a block in the TCA cycle or downstream parts of bioenergetics in PAH. So in summary for the metabolites, we've shown that circulating metabolites do discriminate PAH patients from our controls, and they also predict outcomes, including patients who vasorespond, who will do well on calcium channel blocker therapy, and also survival. Specifically, we've shown increased modified nucleosides, and we've linked that to angiogenin cleavage. We've shown altered energy metabolism, decreased steroid metabolites, which I didn't go into, but also decreased sphingomyelin and phospholipid metabolites, which should be really interesting into looking at how that affects the signaling pathways that they're involved in. And generally, we can say that therapies that correct these altered metabolic pathways may go on to provide effective therapeutic benefit in patients, but that's something that needs to be investigated further. So I could take questions on the metabolites if anyone has any burning questions. Otherwise, I'll move on to the proteomics. So I'll go through the second part. Right, so that was metabolomics. We've also done some proteomic analyses. So the assay that we used in our patients was the somologic assay, and this is an aptamer-based assay. So these are DNA-based molecules which are designed to bind to the specific proteins, a bit like an antibody. And that at the time at which we used it, Somologic had a capability of measuring 1,100 proteins, which we thought was pretty good at the time, but obviously it doesn't quite compare to the protein atlas. But 1,100 is better than other companies that provide similar services. They now offer up to 5,000, but the assay is not open to the same sort of fee-for-service option. So at the moment, this remains sort of the biggest protein data set in patients. So the focus of this really was to develop a panel of proteins that could actually be useful in prognosing patients with pulmonary arterial hypertension. So this is the overall design of the study. So again, we had discovery and validation cohorts. And in the discovery analysis, we were able to identify 134 of the proteins were actually related to survival in these patients by just comparing the survivors and non-survivors. And then we went on to validate 40 of those in the validation cohort, which was split at the point where mortality was close to 50% to maximize our power. Now, to make sure this was statistically robust, we did some reanalysis. We did random sampling analysis. So we picked 5/6 of the patients 18 times to see whether the prognostic ability of these proteins was robust to just removing some of the patients to make sure the results weren't being skewed by a small number of patients. And after repeating this analysis 18 times, we were able to narrow it down to 20 proteins that were really robust in being able to predict survival in our patients. So then we went on to validate this with alternate techniques, because obviously with proteomic assays, we can be fairly confident of the identity of the proteins, but we can't rule out any sort of cross-measurement of different proteins. So we wanted to measure it ourselves with commercially available ELISA and Luminex assays. And we also wanted to compare them with established biomarkers again. So we were looking against BNP, RDW, and 6-minute walk again as our sort of favorite prognostic markers in these patients. So after looking at the head-to-head against BNP, we found that actually 14 of the proteins were independent of these established measures, so they could actually add new information to that which is currently available by the best prognostic marker in PH. And then 9 of those we were able to validate the measurements from the proteomic assay with alternate assays, so the ELISA and Luminex measurements. And in the bottom right, you can just see an example there of one of the assays where you've got a nice correlation between the somologic measurements and the Luminex assay that we did in our hands in the lab. And you can just see that the patients in red there with both assays, the patients who died had the highest levels of this particular prognostic marker. So we narrowed it down from 1,100 proteins to 9 that we think are really robust statistically and also able to be reproduced in our hands in the lab. And what we did then was we wanted to just produce a very simple scoring system. So we did ROC analysis to find specific cutoffs for each marker, which were best related to risk in the patients. And then you can simply count for each protein, how many of those proteins are indicating high risk in those patients to give them a score from 0 to 9. And basically, if you have a very small number of proteins indicating risk in a patient, the survival over time is very good. You can see in blue there, patients with 0 to 2 have a pretty good outcome for this disease, whereas patients with very high levels or low or high risk levels of 6 to 9 proteins, seen in red, have a very poor survival over time. And the intermediate score is intermediate there. And the bottom right just shows the predicted mean survival of patients with the different scores of the panel. But we weren't happy with just creating a model. We wanted to make sure that we could validate that model in a further population. So we approached our colleagues in Paris, who provided a separate cohort of patients. We did all of the measurements again. So we did both the somologic assay and the specific protein and Luminex assays in the lab. And we were able to completely reproduce our findings for the model score. So you can see in the bottom left, the Kaplan-Meier reproduces the effect of the score on survival over time. And what the ROC curves is showing that we're able to show that this model is actually improving on established models. So here we're looking at NT-proBNP, 6-minute walk, RDW, and a combination of those things. But we've also shown that it improves against the REVEAL risk score. So kind of one of the best established clinical risk equations. This model was able to improve on that and actually adds information to that. And again, different levels of the risk score were associated with different estimated mean survival in these patients. And the last thing we've done, we've been able to look at some samples from the group in Gießen in Germany, where they were able to provide us samples from baseline and follow-up, so paired samples from the same patient to look at whether a change in these protein levels is actually associated with outcomes. And what we've actually shown is that if your panel score increases from baseline to the follow-up sample, you actually have an even poorer outcome. So as well as being able to predict baseline risk, you can actually see a change in that score is associated with changing outcomes. The proteins we found are actually quite interesting. They're not completely surprising proteins to have found, although most of them haven't been specifically associated with PAH before. But they do reflect several different facets of the disease. So SD2 reflects myocardial stress. TIMPs 1 and 2 probably reflect the inability of the pulmonary endothelium to repair in these patients. ApoE and insulin growth factor binding protein 1 are related to dysfunction and apoptosis resistance in smooth muscle cells from the pulmonary arteries. We've got complement factors D and H, so it relates to inflammation and innate immunity. Erythropoietin, obviously abnormal iron status we've associated with pulmonary hypertension and outcomes. And reduced plasminogen levels suggest an inability to effectively clear clots in these patients, which is obviously very relevant. So we've identified this protein signature in IPH. 9 proteins in a panel actually perform better than established measures, and we've been able to validate this in an independent cohort from Paris. We've got several challenges remaining that would be interesting in following up. How specific are these changes to pH? Both the metabolites and the proteins would be interested in looking at different diagnoses of pulmonary hypertension. We've started doing that with the metabolites. And we haven't published this yet, but I can tell you a lot of the metabolic changes we see are actually reproduced in different diagnoses of pulmonary hypertension. The tissues responsible for these altered circulating levels of metabolites and proteins, just from a plasma measurement, you don't know where they're coming from. And we're working with our colleagues in Papworth to look at samples taken across the lung and across the heart to see whether their levels change as the blood moves across those tissues, which might help inform us on that. We need to work out if measurement of these metabolites is plausible and useful clinically. So obviously a lot of these assays are very technical and challenging and are currently not available in hospitals. Is that worth pursuing? Should we or can we even target these pathways therapeutically? And in general, we want to be able to integrate these with other omics data. So obviously, I've shown proteomics and metabolomics, but we are generating all these other big data sets, and we want to see how they can best work together. So on that, I'll finish. I'd like to acknowledge lots of different people. We've worked with lots of different collaborators, including the national cohort in the UK, our collaborators in Gießen and Paris, and lots of people in the department who have helped me, in particular Pav Gattori, whose PhD focused on the metabolites. And thank you for listening.
Great, thank you. We'll be happy to take any questions. Marlene?
So that was terrific, Chris. Are your samples on the same patients so that you can make correlations? In other words, if you look at antigen, if you look at a specific pathway, can you track it to an enzyme or something that, you can loosen your criteria and see?
Yeah, I think we can start to look at that. A lot of the proteomic and metabolomic analyses were done on the same patients but sampled at different times because they're from different collections. But there is some overlap that we're going to start looking at. Obviously, the proteins that were selected in the assay are kind of limited to the ones that SomaLogic designed, and a lot of enzymes aren't found circulating particularly highly, so we are kind of limited in that. But obviously, the angiogenin was actually in the SomaLogic assay, and that's where we initially saw that correlation, although we replicated it in-house with some lab assays as well. But that's our next challenge is to really bring these things together and see what we can learn by the combinations of the different data sets.
Right. And your non-disease controls were really a terrific group to work with.
Can you use the microphone?
Sorry. The non-disease controls were a great group to work with. That was terrific. But did you look at that again in the proteomics, and could you distinguish that group together as well?
So the disease controls, I think it's the best comparator group that we have for our PAH patients. We haven't got that data yet in the proteomics. It's one of the things I want to do is to reproduce our panel in a collection of patients with different diagnoses of pulmonary hypertension, including, and plus those controls which don't have pulmonary hypertension, to see how useful it would be if you just screened everyone coming into the clinic. We don't have that for the proteomic data because of the cost. The assay was very expensive when we did it, so we limited it just to the PAH initially. But that is something that I'm planning to follow up on.
And along those lines, do you have the genetic data for all of those patients as well?
We have, for the pulmonary arterial hypertension data, the overlap with the genetics is very good. So we have started to integrate that. So we found variants in the patients which actually alter their levels of the plasma proteins. Okay. And we're going to do that for the metabolites as well. So,
great.
Yeah.
Other questions? We have one question that I'll do my best to read here. Has a little bit of an introduction. In HHT, mid-infrared spectroscopy was used to discriminate HHT from non-HHT. Would this be possible in PAH too? The spectrum was fit into an artificial neuronal network, which learned to discriminate just from blood, serum, or plasma, so no need to know the several metabolites in the blood by name.
I think we could try.
Yeah.
Any new technique coming out, we're always interested in screening it. I think it's definitely worth a shot. If it worked in HHT, it could certainly work in PH.
Absolutely.
That's all I can say.
Yeah. All right. Other questions? Okay. Great.
Great.
Wonderful work. Thank you.